Hi, I am Nidhi Shah, a PhD student in the Computer Science Department at the University of Maryland. My poster is titled Using Scaffolds to Improve Contiguity and Quality of Metagenomic Bins. This is a joint work with Harihara Subramaniam, Jacqueline Meisel, and my PhD advisor Mihai Pap. Increasingly, studies of microbial communities rely on metagenomics. It is done by collecting and extracting microbial DNA from a human or environmental sample, and then high-throughput sequencing is used to capture short sequences called reads. Reads are assembled into longer contiguous sequences called contigs using metagenome assembly tools. Ideally, we would like to have one contig per microbe in our sample, but in reality, that is often not the case. Repeats within and across genomes, uneven abundances of organisms, sequencing error, and strain level variations complicate the assembly process and results in incomplete and fragmented metagenomic assemblies. Therefore, to recover genomes from metagenomic data, these contigs are often clustered into groups such that contigs belonging to the same organism are clustered together. This is called metagenomic binning, and the resulting clusters from this process are called metagenome assembled genomes, or MAGs. There are many methods developed in the community that perform metagenomic binning, such as MetaPad, Concoct, and MaxBin. Most binning methods use information from sequence composition and differential coverage statistics to group together contigs. The basic assumption is that contigs from the same organism will have similar sequence composition and will have similar coverage in a sample. Coverage information from similar environment samples have also been used to reduce false clustering of unrelated contigs. However, these algorithms often miss short contigs and contigs from regions with unusual coverage or DNA composition characteristics, such as mobile elements. What other information can we use that can help cluster contigs from the same genome? We propose that information from assembly graphs can assist current strategies for metagenomic binning. We use Metacaval, a metagenomic scaffolding tool, to construct scaffold graphs, where contigs are nodes and edges are inferred based on linking information such as paired end reads. The scaffolding process can provide information about which contigs can be confidently linked together. Unlike other scaffolding tools, rather than outputting a single linear optimized sequence for each scaffold, Metacaval preserves strain variants and identifies subgraphs as scaffolds. To use scaffolds with existing metagenomic binning methods, we needed a way to accurately estimate scaffold coverage. So for each scaffold component from Metacaval, we assign coordinates to each contig based on the distance estimates from the edges and the contig orientation in the scaffold graph. Once all contigs have coordinates relative to the first contig in the scaffold, we compute per base depth of coverage for the scaffold. We also have a misscaffolding detection subroutine that breaks up scaffolds when median coverages of the two groups are vastly different. For our experiment, we took 20 complex stool samples from the Human Microbiome project. We ran MetaPad with contigs and MetaPad with our scaffolds. The plot on the left shows the cumulative sum of bins sorted by decreasing order of bin length. For all samples, there was an increase in the number of contigs and length of the overall bins when using scaffolds instead of contigs for binning. We used CheckM to evaluate completeness and contamination of bins. Binning with scaffolds improves completeness of the bins while still having majority of the bins with low contamination error. In summary, scaffolding and binning have often been used as separate analysis to improve metagenomic assembly quality, but we show that using scaffolding information, specifically scaffolds from Metacaval that preserve strain and structural variation, can help improve the contiguity and quality of bins. This is still preliminary work, and we believe that additional algorithmic improvements can further boost this benefit. Thank you.